Hey, welcome back. We're going to continue on with the chapter on chemistry. This is chapter 2. We have been covering the organic molecules. We did lipids already, and we just finished up carbohydrates. And now we're going to the third macromolecule that we're going to be covering in this class. And that is the molecules we call proteins. And again, remember, fats, carbs, proteins. These are the three macromolecules that people generally worry about when they're concerned about their diet. So how are proteins different from lipids and carbs? Well, they have a lot in common with lipids and carbs. Remember, lipids are primarily carbon and hydrogen. There's a little bit of oxygen involved, but carbs are carbon, hydrogen, and oxygen. Remember the carbohydrate, the water, carbon, water. What's new here is proteins have nitrogen. It's part of an amino group. There are some sulfur molecules on some of the side chains, but the real most common atoms are carbon, hydrogen, and oxygen, and nitrogen. This is really key. And it's important because the amino groups that the nitrogen is a part of are a part of every single amino acid. It's why they're called amino acids. All right, so what do they do? Proteins have a variety of jobs to do in the cells. One is they provide structure. If you want to think about some proteins that you already know about, like in muscle cells, actin and myosin, really important for like holding the shape of the cell. They act as enzymes, and I'm going to remind you what that means. A catalyst is something that is present and allows chemical reactions to happen more readily, but it itself is not involved in the reaction permanently. I'll give you an example. An analogy, let's put it that way. Two people might want to get married and they can go together to a justice of the peace or some other legal authority or religious authority, and then that third person can marry them, join them to each other, but that third person is not now part of the marriage, right? I mean, it catalyzes it, makes it happen more quickly. Those two people could have lived together and eventually become common law spouses, but it went faster when they involved this catalyst. And the term enzyme, all it means is that it's a catalyst that happens to be a protein. That's what enzyme means. Okay, regulation of the cell cycle, for example, transportation, moving things around, moving the cell around in space. And in defense, we have the antibody proteins that help us fight off infections. And offense, there are molecules that are toxins that are used to fight off other microbes, or microbes can fight with each other. Okay, so lots of different functions for proteins. They are all of the proteins are made of individual subunits, just like we said, the polysaccharides, the complex carbohydrates, you made up of a lot of sugars. Proteins are made up of a lot of amino acids. Those are the individual units, monomers. There's only 21 different amino acids, and most organisms use only these 21 amino acids. Let's see. I'm going to show you on another slide. There are 3 parts to every amino acid. 2 of them are exactly the same in every single amino acid. There's an amino group and there's a carboxyl group, and those are exactly the same. And that's how they connect to each other. And then this side chain, that's what's different between the 21 different amino acids. Some of these side chains or side groups, some of them are hydrophilic and some of them are hydrophobic. And so the hydrophobic ones like to hang out with each other, and the hydrophilic ones like to hang out with each other. And that determines how this thing is going to fold. I mentioned that the amino acids, they have these 3 parts, and then they kind of connect to each other. So then I guess I should do it this way. The amino group on this one is going to connect to the carboxyl group on this one, and that's how they hold hands in a chain. And what we call that connection between two amino acids next to each other, that's referred to as a peptide bond. I know this is all terminology, but make sure that you know these terms. I want you to know amino acid. I want you to know the three different parts of the amino acid, and I want you to know that the bonds between different amino acids are known as a peptide bond. All right, so here's a cartoon of some amino acids. This is the generic version of it. There's an amino group. That means it's a nitrogen with a couple of hydrogens hanging off of it. There's this central carbon. It's referred to as the alpha carbon. Not like it's dominant. It's like the central one. We would start counting carbons from there. And then the other end of the amino acid is this carboxyl group. So the amino group gives it the name, and then the carboxyl group, because it can give up this hydrogen ion, it's an acid. So that's why it's called amino acid. All right. Then attached, the other things that are attached to this central carbon, this alpha carbon, there's always a hydrogen ion here. Note they all have that in common. But then the other side, the carbon, is attached to something else. And these are highlighted in blue. And it could be as simple as another hydrogen ion. That's a particular amino acid called glycine. It could be a much more elaborate thing, this ring structure of carbons and hydrogens. This is phenylalanine. For the purposes of this class, you're not going to have to know all of the individual amino acids, but what I'm trying to get across to you is a particular amino acid is shaped a particular way because of this R group. Cysteine, so here's an example of one that it contains sulfur. Okay, so you get the idea. Some of these are going to interact better with each other than others. So for example, this one looks like it's going to be carbons and hydrogens. That's going to be hydrophobic. This one includes some carbons and nitrogen and oxygen. That thing's going to be like not sharing the electrons equally. So that thing's going to be polar. So hydrophilic. So these are not going to interact with these very well. And so they'll repel each other. And that's going to determine the shape of this whole molecule. All right, so the peptide bonds. So I mentioned that the amino acid number 1 is going to bind to amino acid number 2. They all have an amino group. So here's on the left of this one, they represented it in one way, but it's always the same. It's a nitrogen and a couple of hydrogens, just like here, a nitrogen and a couple of hydrogens. And then the carboxyl group is this carbon, a double bonded oxygen, a hydroxyl group, and it's represented here with the same carbon, an oxygen, and then the hydroxyl group. The point is that the carboxyl group will bind to the amino group of the other amino acid. That's how they hold hands. And just like we did when we put like multiple glucoses together, or a glucose and a fructose, you get rid of a hydroxyl group and a hydrogen. That, my friends, is H2O. So you release a molecule of water. So that's referred to again as a dehydration synthesis. You're putting two things together and a water molecule is coming out. And then that bond that forms here between the carboxyl group and the amino group right there, that's referred to as a peptide bond. So when you put a couple of these amino acids together, that's a dipeptide. If you have a long chain, that's a polypeptide. Amazing. Okay, and then finally, the most important part of proteins is they're all made of these amino acids, right? They're always one long chain. Well, they're always a chain of amino acids. If it's long enough, this thing can fold and take certain shape. And so the order in which the different amino acids appear, that's referred to as the primary structure. So you have a glycine and then a phenylalanine and then a different one. And then that's the primary structure. And then that thing, that primary structure, has some sections on it that might do like a curlicue or a zigzag. These are referred to as secondary structure. You may remember hearing about these, alpha helix or beta pleated sheet. And then on top of that, We have some additional folding that happens. So you keep the alpha helix and the beta pleated sheets, but then on top of that, there might be some hairpin turn that, these things will line up together. That's referred to as tertiary structure. So it's like the third level of shape to this thing. And then finally, if you have more than one polypeptide, if you have more than one chain that has to come together to form a functional protein, that's referred to as quaternary level structure. So the fourth level of structure. The shape of the protein determines what job this protein can do. Just like if you think about, and more analogies, if you think about like a can opener, if you've ever worked with a can opener, it's got to have the handles, and then it's got to have the blade that sticks into the can, and then there's got to be the thing that you turn so that this can move. But if any part of that is messed up, like if that blade, it's there but it's at the wrong angle, you've got all the parts, these, like the primary structure might still be fine, but if my tertiary structure is messed up, that protein's not going to work right. And these proteins are really sensitive to changes in pH, for example, or changes in temperature. Those proteins denature, they change shape. And although the primary structure stays the same, we lose the overall tertiary structure or quaternary level structure. And then that protein might not work right. This will come up later. All right, so this is the end of part 3. I'll stop there and I'll see you over in part 4.